Okay, it's Malcolm and good morning everyone. Today we continue our lesson with tRNA, rRNA, and then proceed with transcription and translation. Now, tRNA is transfer RNA. Okay, it is a molecule that consists of a single RNA that is made by transcription of the DNA in the nucleus. Okay, I will further explain what is transcription. Now, first thing that you need to know is transcription of the DNA in the nucleus will give out or synthesis RNA. RNA, when I say RNA, it can be tRNA, it can be mRNA, it can be rRNA. Right? Now, the single RNA strand of the tRNA is only 80 nucleotides long. Okay, meaning to say it has only 80 nucleotides bases. Then it folds back on itself, right? And form a molecule with 2D structure. Okay, now in the notes, it was stated there three-dimensional structure. So I want you to make the proper correction. Okay, it should be 2, not 3. Okay, 2D structure because of the presence of complementary stretches of nucleotide bases that can hydrogen bond to each other. Refer to the diagram under these notes. There are 4. You can see that there are 4 diagrams there that represents the tRNA. One is a 2D structure, and then the 3 is a 3D structure, if I'm mistaken. And then this tRNA, it can twist and folds into a compact 3D structure that is roughly L shape. Okay, the loop extending from one end of the L includes the anticodon. The L-shaped molecule has 2 functional ends. Okay, it has 2 functional ends, the acceptor end and the anticodon loop. These are the 2 functional ends. To be precisely, there are 3 ends. Okay, the 3 prime, and then the 3rd prime, the 5th prime, and the anticodon loop. But functional ends only the 3rd prime end and the anticodon loop. The 3rd prime end is the acceptor end. It is also known as the acceptor end. Okay. Okay, again, as what I've mentioned earlier, it has 3rd prime and 5th prime ends. Okay, the 3rd prime end, okay, it is specific amino acid is attached to the 3rd prime end. Meaning to say, at the 3rd prime end, specific amino acid will be attached there, right? And this end is known as the acceptor end. And then the 5th prime end, okay, it is a nucleotide triplet that can best pair with the complementary codon on mRNA. The bottom region has complementary anticodon, which I mentioned earlier, that form hydrogen bonds with the mRNA codon by complementary base pairing. Do you get that? Meaning to say, the anticodon, okay, in the tRNA, it will form hydrogen bond with the mRNA codons, okay, by complementary base pairing. You know complementary base pairing? If the loop has a sequence of GGG, okay, not the loop, the anticodon on the tRNA. If the sequence there is GGG, then it will form a complementary base pairing with CCC on the mRNA codon. Right? Remember, on mRNA is codon, the genetic code is called codon, and on the tRNA is anticodon. Now, the function of tRNA is to transfer amino acids from the cytoplasm, okay, to a growing polypeptide in a ribosome. Can you imagine that? tRNA, it carries the amino acid from the cytoplasm into the ribosome in the cytoplasm as well. Okay, so it transfers the amino acid from cytoplasm to the ribosome. Each tRNA is specific for a type of amino acid. So meaning to say every tRNA will only carry a certain amino acid that is correspond to the anticodon. Okay, it's anticodon, the tRNA anticodon. Example, tRNA ALA, like what you see here. Okay, tRNA, ALA, it carries alanine, and tRNA, MET, it carries methionine, right? Okay, this is the 2D structure of the tRNA, which is it has the shape of a clover leaf. Okay, and then this is the interaction, the hydrogen bond between the nucleotide bases. You can see it's here, and it has 80 nucleotide bases altogether. This is the 3D structure. Okay, it twists and folds. Remember I mentioned earlier, to form a 3D structure. Okay, this is another 3D structure, how it will look like. Okay, this is in its icon shape. Okay, you can see the acceptor end here, which is the third prime, and then the anti-codon end here. Two functional ends. Okay, you can see here the third prime end, it forms an ester bond with the amino acid that it carries. Okay, ester bond. Do you have this diagram? I can't recall. I don't think so. But take note that the third prime end forms an ester bond, okay, with amino acids that it carries. Again, the amino acid that it carries here correspond to its anticodons here. Okay. Okay. Now you have this diagram, this table in your module. So I give you 10 minutes or 15 minutes. Take your time. Okay. Pause this video. Try to fill in, okay, the correspond genetic codes as well as the amino acid that codes coded, okay, in this table. Like if you have codon on mRNA is AUG, okay, and AUG, you know, is a start codon, so you put, so it's start here, right? So what is the correspond the triplet code of DNA here? Okay, as well as the anticodon on tRNA. If it's AUG, then you get, this should be T, okay, uracil pair with adenine, okay, T, A, G, and C, right? So I give this example. Now the others here, you do it on your own. Okay, 10 minutes from now. Okay, pause the video. Okay, see you later. Bye. Okay, done. All right, let's check your answer. All right, so this will be your answers. Okay, so TSC, okay, like what I've mentioned earlier, it codes the start codons, right? Start codon, which will bring the methionine amino acids. And then ATT, ATC, ACT, these are all stop codons. So here you should write stop as well. S, T, O, I, susahnya tulis P, right? Okay. And then here are all stop because it correspond with this, complement with this, right? Betul kan? ACT, you get UGA, right? Remember, in RNA, there is no thymine. Thymine is replaced with uracil. Okay, now, these stop codons, like what I mentioned earlier, all right, there is no tRNA that correspond with stop codons. Okay, so here will be no tRNA with corresponding anticodons, or there is no anticodons that correspond with stop codons. Okay, no corresponding no anticodons correspond with stop codons. Get it? You can write either no tRNA with corresponding anticodons or no anticodons corresponds with stop codons. So since you don't have any anticodons, okay, that correspond with stop codons, So the tRNA will not code any amino acid or will not transfer or carry any amino acid. Okay, here should be written as no amino acid. Please write here. Okay, this box here, no amino acids. Okay, so I guess the other would be easy for you. Okay, so these are the answers. Refer on your own. And then the coded amino acids here should be alanine and cysteine. All right. Okay, done. All right, nice. Let's proceed. Now, ribosomal RNA, rRNA. Okay, it is a molecule made by transcription in the nucleus. Okay, similar with mRNA and tRNA. Exported to the cytoplasm to form the ribosome. Okay, ribosomal RNA is the most abundant type of cellular RNA. Ribosome made of proteins. Now we are talking about ribosomes, right? Ribosome made of proteins and one or more rRNA. Like what you have learned in the SEM 1. One ribosome consists of proteins, okay, that made up the large and small subunit, and then it consists also with one or more rRNA. Adds each amino acid brought to it by tRNA to the growing end of a polypeptide chains. Okay, and ribosome consists of small subunit and the large subunit. The small subunit is the binding site for mRNA, and large subunit is the binding site for tRNA. Okay, look in the diagram of the ribosome. It's there in your module. Okay, I'm sure you are able to identify which one is the small and large subunit of the ribosome. Now, in eukaryotes, the subunits are made in the nucleolus. Okay, because when it comes to eukaryote and prokaryotes, it's slightly different. Okay, and eukaryotes ribosomes are more complicated. Now we focus with eukaryotes. Okay. Ribosome has a binding site for mRNA, okay, a binding site for mRNA, and 3 binding sites for tRNA. Okay, the binding site for mRNA can be found on the small subunit, whereas the 3 binding sites for tRNA can be found on the large subunit. The 3 binding sites for tRNA, okay, they are known as PAE site. P site because it's peptidyl tRNA binding site. Okay, peptidyl tRNA. A site is amino acyl tRNA, and E site is exit site. Do you need to know this term here? Yes, you do. Okay, it's quite fancy. Okay. Now, the P site will hold tRNA that carrying the growing polypeptide chain. And then A site, it holds tRNA carrying the next amino acid to be added to the chain. E site is where tRNAs that have delivered their amino acids to the growing polypeptide chain exit the ribosome. So from left to right sequence, it will be EPA. Okay. Look at the diagram. Okay, E is the exit where the tRNA that already delivered the amino acid will exit the ribosome, and then P site, okay, the site where, okay, it holds the tRNA that carrying the growing polypeptide chain. A site is the site where the tRNA carrying the next amino acid to be added to the chain. So A site macam baru sampai lah, tRNA that just arrived in the last subunit of the ribosome, it stays there. Okay, any new arrival of tRNA at the last subunit will landed in the A site. Okay, so after it landed on the A site, it will proceed to the P site and then finally to the E site. Okay, Okay, now this diagram here to show you a 3D model of the ribosomes with the mRNA and tRNA, okay, inside each of the EPA sites. Okay, you can see the large subunit of the ribosome, the small subunit. Okay, this is the mRNA. Okay, class, always remember reading of the mRNA is from the third end to the prime end, right? The direction of translation. And then you have this diagram, right? Okay, this diagram just to show you the EPA and such. Okay, all right, I already explained this to you. Next, okay. A site where you have the tRNA that just arrived with its corresponding amino acids. The P site, you have the tRNA with the long polypeptide or growing polypeptide chain. Okay, the E site where the tRNA exits the ribosome. Okay, 2013. An anticodon is a sequence of nuclei on the. What do you think the answer is? Alright, it's B. Pretty obvious. Okay, now the big deal for today. Okay, synthesis of protein, which is transcription and translation. So now all this while I've been saying about transcription and translation. Now transcription and translation, okay, it means synthesis of protein, how protein is being synthesized by your body cell, okay, in eukaryotes as well as prokaryotes, how they synthesize their own protein. It's the same thing by transcription and translation. So from this diagram here, you can see that transcription will produce or synthesis one RNA. Okay, transcription. Then from translation, the RNA, from the RNA you get a protein. Okay, that's the basic knowledge of transcription and translation. All right, now let's proceed. Transcription. Transcription is the synthesis of RNA from a DNA template, the leading strand. So what happened? The stretch of DNA that is transcribed into RNA, okay, transcribe ni tepek lah senang cerita. Stretch of DNA that is transcribed into RNA is called the transcription unit. So meaning to say any part or any stretch of the DNA that is being transcribed, that is being read to synthesis a RNA is known as transcription unit. Most RNA synthesis, okay, require one of three RNA polymerases. Okay, enzymes present in all cells. Meaning to say, every cells, okay, in eukaryotes, they have three RNA polymerase. Three types of RNA polymerase. Each one of these has different function. Okay, but for your level, it's sufficient for you to mention only RNA polymerase. Okay, now RNA polymerase 1, it catalyzes synthesis of rRNA. RNA polymerase 2 catalyzes synthesis of mRNA. And RNA polymerase 3 catalyzes synthesis of tRNA and one of the rRNA molecule. Right? So how are you going to memorize that? I don't know. If you can memorize this, better. Okay, it might come out. I don't know, but it's not mentioned in your syllabus anyway. RNA polymerase require DNA as a template and have many similarities to the DNA polymerase. Remember, you have learned about DNA polymerase. Now, similar or like DNA polymerase, RNA polymerase carry out synthesis in the 5' to 3' direction. Okay, so synthesis of RNA is from the 5' to 3' direction. The RNA molecule being synthesized at the 5' end, where addition of nucleotides towards the 3rd end. until the molecule is complete. Can you picture that? This is the RNA. Okay, this is the fifth end here. All right, it starts with the fifth end, and then addition of nucleotides towards the third end, because when the strand is about this short, the third end is somewhere around here. All right, and then keep on adding the nucleotides, and initially you get the third end over here, right? And then let's say it wants to grow, so another addition of nucleotides is added here. Now you tell me, where is the nucleotide being added? It is towards the third end. That's why towards the third end. Okay, until it forms a complete molecule. Now, transcription consists of initiation, elongation, and termination stages. There are 3 stages in transcription. So you might want to add here stage. Okay, stage. Right, stage. Okay, let's go to the first stage, initiation of transcription. So what happened here? Okay, so what you can see in this diagram here. Where's my pointer? All right, so you can see this diagram here. Okay, this RNA polymerase. Okay, sorry, this is the RNA polymerase. It attaches to the promoter site. This is the promoter site. There is a promoter site on the DNA. Okay, you can see the promoter site here on both strands of the DNA. So RNA polymerase attaches to the promoter site. Okay, and the RNA polymerase will unwind the DNA to initiate transcription. Okay, what happens when the DNA is being unwound? This will expose the bases of the transcribing DNA strand. Right, now in the transcription of the DNA, if you still recall, okay, DNA helicase will unwind the double helix, right? Then DNA polymerase will come in and start the transcription. But this is different with the transcription of RNA. Now, RNA polymerase moves past the promoter, okay, to begin transcription of the protein coding sequence of DNA, because the promoter is not being transcribed. So RNA polymerase come here, okay, then just move, oh sorry, it comes here and then it causes the double helix to unwind, and then the RNA polymerase just pass through the promoter site to start initiation of transcription. Okay, sorry, to initiate transcription, not to start, initiate transcription. To initiate transcription. We don't use start because start is not scientific. Okay, and in transcription of protein, they want the specific word for that, which is initiate. All right, elongation of transcription. Now, what happens here? RNA polymerase. Okay, sorry class, this polymerase P is a small letter P. All right, RNA polymerase will move along the cistron on the DNA. Okay, cistron of the DNA is the transcription unit in the 3' to 5' direction, as you can see here. 3' to 5' direction. Okay, and passes complementary nucleotides to the transcribing DNA strand one at a time to build up RNA. Okay, RNA or mRNA. I put here mRNA to give one example. Okay, again, it depends on the RNA polymerase. If type 1, you are synthesizing rRNA strand. Okay, now so since I'm talking about general RNA, okay, the same goes here in your exam. If they don't specify which type of RNA polymerase, okay, or which type of RNA, so you might as well just use RNA polymerase. And here you are transcribing a RNA. Okay, Now, if you want to know where does the nucleotides available, they come from, they are available in the nucleus. They are there. Now, the first nucleotide at the 5N prime, at the 5N, okay, of a new mRNA chain initially retains a triphosphate group. Each additional nucleotide is added at the 3rd prime end of the growing mRNA molecule, like what I mentioned earlier. The last nucleotide to be incorporated or added will have an exposed 3rd prime hydroxyl group. Okay. RNA polymerase moves on to another region of the transcribing DNA and causes the zip at the promoter site to close and the double helix reform, as you can see here. Okay, when the RNA polymerase already passed through the promoter site, it goes to another region. The promoter site region will close. Okay, and this will make the double helix reform. Elongation of mRNA continues until termination. Okay, apparently this is the termination site. Now, termination of transcription or termination stage. What happened? RNA polymerase recognize a termination sequence. Okay. Now, the RNA polymerase will detach from the DNA, from the template DNA and the newly synthesized mRNA. So, as you can see from this diagram, this RNA polymerase, it detaches from the template DNA and the newly synthesized mRNA. So apparently earlier this RNA polymerase is on these two molecules here. Now it detaches itself, okay, from the template DNA and the newly synthesized mRNA. Now class, this newly synthesized mRNA or newly synthesized RNA is known as pre-RNA. Okay, if you are talking about synthesizing of rRNA, so this is a pre-rRNA. Okay, now to be general, this is a pre-RNA. Okay, now since I'm giving out example of mRNA, so this is a pre-mRNA. Then the pre-mRNA are modified before moves into the cell cytoplasm for translation. Class, the modification of this pre-mRNA, it occurs very quickly. Okay, because the process of transcription and translation, it occurs simultaneously. The pre-mRNA consists of complementary introns and exons. Okay, because during transcription, okay, both introns and exons on the DNA is being transcribed. So the mRNA will have both introns and exons. Introns is nucleotide bases that do not code for any protein. Okay, and exons are nucleotide bases that codes protein synthesis. So the introns are cut out while the exons are spliced together. Splice ni dicantumkan, digabung, splice, okay, through post-transcriptional modification and processing activities to produce mature mRNA or mature RNA, okay, for transport and translation. Okay, I believe you have the diagram about the post-transcriptional modification and processing in your module. Right? You can see the difference there. Now, this chart here, just to show you the differences between a DNA and mRNA. Not really mRNA, it's RNA. Okay, RNA. The differences between RNA and DNA. Okay, I'm sure you know already. You have learned this in the first semester. Okay, this diagram here to show you the intron and exon. So we say on your DNA, there are many introns and exons. That's what you can see. Okay, yeah, from the DNA, okay, you get this mRNA. So since the DNA has exon and intron, the same apply to your mRNA or RNA. It has exon and intron. All right, and then finally you get a mature RNA, which will exit the nucleus towards the cytoplasm for translation. Okay, this video, okay, I'm going to play it now. All right, if you feel tired, you may take a rest. Now this video will explain to you about translation. Now we are going into translation. Okay, and we already our lecture is already um, ran for 30 minutes. You may pause a while, take your rest, pause the video. All right, take 5. Or you want to continue also can, no problem. Okay, now let's proceed with this video. How do I play this video? Aish. Okay, you know what? I will share the video in the group later, in the GCR later. All right. Okay. Now translation. It is the synthesis of a polypeptide using the genetic information encoded in a mRNA molecule. Or simply, translation is a process where codons in the mRNA is read to synthesize specific polypeptide. Okay, now class, before you proceed with this part of this video, I advise you to watch that video earlier first about translation, so that you have an overview of what is translation all about. Okay, now already watched the video? Alright, now let's proceed. Before translation, okay, prior to translation, now what happened? The tRNA is being referred to a non-charged tRNA because it has not bind with any amino acid. Okay, so this tRNA in the cell cytoplasm, it must be charged. Must be charged means it must bind with amino acid that correspond to its anti-codon. Okay, now, so what happened? This tRNA, okay, amino acid tRNA synthase, okay, coupled with ATP, as you can see from this diagram here. This amino acid tRNA synthase or synthetase, both can be accepted. Okay, it will bind tRNA to amino acid to form a charged tRNA, and a charged tRNA is known as amino acid tRNA. Now you know already why the A site in the ribosome is known as A site or amino acid tRNA sites. Okay, it is for the amino acid tRNA to bind with. Okay, so you can see here how this amino acid synthetase, okay, it catalyzes the reaction, okay, of amino acid with the amino acid tRNA synthetase catalyze, okay, the transfer of amino acid into tRNA, okay, and it needs ATP. Here finally you have the tRNA with the with its correspond amino acids. Okay, and this diagram here, it tells you that the charged tRNA dissociates from the amino acid tRNA synthetase. Now, why this process is needed? This to make sure the charged tRNA carries the correct amino acids specified by its anti-codon. Right? Now, example is threonine is carried by tRNA with the anti-codon UGA or UGG or UGC. Okay. Okay, this is the diagram that you have, right? Because the purpose is I want you to read the descriptions here. Right? Okay. do you need to write this descriptions here in the exam? No, it's not in your syllabus. Okay, this is just for your extra knowledge. Okay, now translation occurs in the ribosome, transcription occurs in the nucleus, and translation occurs in the ribosome in the cell cytoplasm, and divided into 3 stages. Okay, initiation stage, elongation stage, and then termination stage. Okay, now in initiation, okay, first initiation factors, you can see these initiation factors here, right? It will attach to the small ribosomal subunit. Okay, this is the small ribosomal subunit. Okay, and the small ribosomal subunit binds to the mRNA at the AUG start codon. Can you see the sequence here? Initiation factor, initiation factors attached to the small ribosomal subunit, and then the small ribosomal subunit will bind to the mRNA at the AUG start codon. Next, the tRNA that bears the first amino acid of any polypeptide is the initiator tRNA. Okay, and this initiator tRNA, it carries the amino acid methionine, which correspond to its start codon. Okay, thus the first amino acid on new polypeptides is methionine, right? Why methionine is the first amino acid on polypeptide? Okay, because ribosomal subunit will always bind to mRNA at the AUG start codon, and when this happens, tRNA that carries the first tRNA, okay, or the initiated tRNA is tRNA that carries methionine. Because why methionine? Methionine will complement with the AUG start codon on the mRNA. The tRNA binds to the start codon, and one of the initiation factor is release. So the moment this initiated tRNA, okay, which is a tRNA with methionine, bind to the AUG start codon, one of these initiation factor will be released. Okay. Now, if methionine is not needed as part of the final polypeptide chain, it will be simply removed after the completion of the polypeptide synthesis. Now, the large ribosomal subunit will bind to the small ribosomal subunit, and the remaining initiation factors are released. The initiation complex is complete with EPA sites. So now, the large ribosomal subunit, okay, will bind to the small ribosomal subunit. Then what happens after that? The initiation factor will detach or release. Okay, when this initiation factor is already released, initiation complex is complete or formed. So this whole thing here is known as the initiation complex. Some books refer this initiation complex as ribosomal complex, something like that. I'm not sure whether that term is a proper term for that. Okay, but initiation complex is a proper term, okay, for this complex. So an initiation complex consists of small subunit of the ribosome, the large ribosomal subunit with the tRNA and the initiator tRNA. Okay. Alright, next, after initiation stage is the elongation stage. So elongation, okay, is a stage of translation in which amino acids are added one by one to the growing polypeptide chain. as you can see from this diagram here. So the appropriate amino acid tRNA will recognize the codon in the A site and binds by base pairing. This is the base pairing here, okay, of its anticodon with the complementary mRNA codon. So sorry, it should be from here to here, right? Complementary base pairing. So let me rephrase that. Appropriate amino acid tRNA recognize the codon in the A site and bind by base pairing of its anticodon with the complementary mRNA codon. This binding step requires several proteins, okay, and these several proteins are called elongation factors, as well as a GTP. Okay, so for this amino acid tRNA to bind with the complementary codon on the mRNA, it needs GTP as well as elongation factors. Now, after that, what happens? The amino group of the amino acid at the A site, okay, is now aligned with the carboxyl group of the preceding or previously amino acid that is attached to the growing polypeptide chain at the P site. Okay, can you get that? Amino acid of the amino acid tRNA in the S site is aligned with the carboxyl group of the previously amino acid chain on the P site. Or if in the P site there is the methionine, so what happened? The amino group of the amino acid at this A site will be aligned with the carboxyl group of the methionine, okay, at the P site. Okay, next. Remember earlier I told you the initiated tRNA will land at the P site, okay, and then the subsequent amino acid tRNA will land at the A site. A peptide bond forms between the amino group of the new amino acid and the carboxyl group of the preceding amino acid. Okay, that's what you can see in this diagram here. New amino acid. So the strand of polypeptide in the P site will shift to the A site. The polypeptide is released from tRNA at the P site and attaches to the amino acid tRNA at the A site. Next, ribosome moves one codon at a time towards the 3' end of mRNA. You can see here. Okay, the ribosome moves one codon at one time. And when this happens, the growing polypeptide chain is transferred to the P site. So earlier it was on the A site, now it's transferred to the P site. Okay. Why is being transferred to the P site? Because the tRNA which was on the P site will be transferred to the E site, and then the tRNA which was on the A site together with the polypeptide chain is transferred to the P site. That's why you have the polypeptide here, which was initially on the S site, it's shifted to the P site. Okay, because it is this polypeptide here was attached to the tRNA on the S site. So since the tRNA on the S site shifted to the P site, so it carries together the polypeptide. It carries together the polypeptide and transfer the polypeptide from the S site to the P site. Okay, the uncharged tRNA. Okay, uncharged tRNA. That's what I explained earlier. The tRNA without amino acids is moved from the P site to the E site and exits the ribosome to join the cytosolic pool of tRNA. So meaning to say the uncharged tRNA, okay, it will leave the ribosome and it goes to the cytoplasm cell. Now when it reaches the cytoplasm of the cell, what happens? It will be charged back, okay, to form amino acid tRNA. Ribosome ready to accept another amino acid tRNA. So another amino acid tRNA will arrive at the A site, okay, and the elongation continues. Now, termination, the final stage of translation. Now, when the ribosome reaches a stop codon, okay, that's what you can see here. When the ribosome reaches stop codon, what are the stop codons? UAA, UAG, and UGA. The A site will bind to a protein release factor. So this is the protein release factor. The moment the ribosome reaches the stop codon of the RNA, this release factor will come in and sit in the S site. Not sit, sorry. Binds to the S site. Okay. The release factor will hydrolyze the bond between the polypeptide chain. Okay. And the tRNA, causing the release of the polypeptide chain from the tRNA molecule in the P site. So means say when this release factor bind to the S site, it will hydrolyze the bond between the tRNA and the polypeptide at the P site. Okay. So what happened? The polypeptide chain is released. The hydrolysis reaction, okay, also separates the translation complex into mRNA molecule, the release factor, tRNA molecule in the P site, and the large and small ribosomal subunits. So meaning to say, let me rephrase that. Okay. Okay. Release factor arrive at the S site. It will hydrolyze, okay, the bond between the polypeptide chain and the tRNA at the P site. And when this happens, the polypeptide chain is released, as well as the ribosomal subunits, the large and small subunits dissociates. When that happens, the release factor are released, as well as the mRNA and tRNA at the P site. Okay, so that is about translation. This diagram here to show you the comparison between transcription and translation in eukaryotes and prokaryotes. Prokaryotes such as bacteria. Okay, their transcription apparently, okay, there is no introns and exons on the RNA because their DNA, okay, do not have introns. Okay, every gene in their DNA codes protein synthesis. Okay, unlike the eukaryotes, there are some introns here. Okay, so what happened? RNA processing, okay, to remove the introns and splice the exons. Because introns not really at both ends. It can be at both ends together with the middle part here as well. So if these introns are already removed, what happens to the exons here? It needs to be combined, right? So it needs to be spliced. The word here is splice, not combine or glue or whatever. Splice, right? Okay, these are several types of RNA that can be found in eukaryotic cells. There are many. Okay, so you read on your own. I'm not going to explain this. Okay, the ones that is important for you is the mRNA, tRNA, and rRNA. Okay, so test your knowledge. There are quite a bundle of questions here. Okay, so once transcribed, eukaryotic mRNA typically undergoes substantial alteration that includes after transcription. What happens to mRNA? I already explained earlier. Okay, the introns and exons. Okay, fusion with, sorry, excisions of introns. Okay, introns are significant to biological evolution because why? Why introns are important to evolution? Because their presence allows exons to be shuffled. Okay. Now, class, to cut short the time, I won't explain every bit of the question. All right, I just show you the question. Assume, okay, you pause the video now. You do all these questions first. Okay, you do all these questions, then you come back to this video to check your answers. All right, because I want this one to save the time. Now it's already 50 minutes. I've been talking for 50 minutes. Our class, our lectures have been running for 50 minutes. I just want to minimize the size of the file later on to be uploaded actually. Okay, so these questions, what do you think your answer is? Okay, check your answer. All right, this is the answers. If you don't understand why these are the answers, always you can ask me. Okay. Oh my, needs to be charged. Oh. Okay. All right. Okay. Okay, so the answer is here. All right. Okay, the answers are, okay, if you couldn't find the answer, look back at the module, it's there. Okay, most abundant type of RNA is the rRNA I mentioned earlier. Okay, and then this diagram, what do you think the answer is? Okay, answer is D. And then this question, okay, the answer is B. Right. Okay. Right. Right. You get that? Okay. All right. All right. Okay. Okay. Right. 2015 big paper. Okay. This is the answer. All right. Okay. Okay. This is the answer. Right. Okay. So that's it for today. All right. Thank you so much. Okay, anything you don't understand, please ask us in the group. Okay, thank you.